A preparation of olfactory epithelium is mounted on a dish and placed into a stream of humidified air under a dissecting microscope. A reference electrode is electrically connected to the tissue through a buffer solution. A recording electrode is placed on the surface of the olfactory epithelium. The olfactory epithelium is stimulated by computer-controlled odorant puffs administered into a stream of humidified air, which carries the odorants to the olfactory epithelium. EOG signals are transmitted through an amplifier and digitizer to the recording computer.
Hi, I'm Katherine Cygnar from the laboratory of Haiqing Zhao at the Department of Biology at the Johns Hopkins University. Today we will show you a procedure for recording the electroolfactogram from mice. We use this procedure in our laboratory to study the responses of olfactory sensory neurons to odors. So let's get started.
The recording apparatus consists of a recording electrode, reference electrode, air delivery tube, specimen stage, and dissecting microscope, all anchored onto an air table within a Faraday cage. Micromanipulators are used for placement of the electrodes in the air delivery tube. A continuous airstream is bubbled through distilled water to add humidity before passing through the air delivery tube and over the specimen. A 60 mm culture dish filled with Cygnar to a depth of 6 to 8 mm is used as a mounting surface for the specimen. A well and a channel are hollowed out of the Cygnar in the mounting dish to provide a means to electrically connect the reference electrode to the specimen via modified Ringer solution.